A very good afternoon to one and all. It's my immense pleasure to welcome you all for the two-day international webinar on current scenario in therapeutics, prospects and challenges organized by Departments of Life Sciences, Srimad Andavan Arts and Science College, Trichy. A good start with a positive attitude is the most important step of journey to discover more opportunities. Let's start the program with prayer.
वन्दामहे श्रीमदाण्डवन् पादुकाङ्कम् श्रीरङ्गक्षेत्रपवित्रोज्ज्वलपारिजातम् यत्र मानवाः सर्वे सन्मार्गदर्शिताः नित्यं पठन्ति ते विज्ञानकलाभाषाः पाठातिरिक्तविषयेऽपि प्रवृत्तास्ते सुशीलगुणावपि अनुचरन्ति नित्यम् नानाकलाक्रीडेष्वपि सुशोभिताः सर्वत्र सर्वदा ते आत्मप्रभावैः ओषधिवने स्थापित प्रयोगशालासु सम्यक् प्रकाशयन्ति ते स्वप्रभावम् अत्रैवाधीतयन्त्रविद्यासु निपुणास्ते विदेशेऽपि उज्ज्वलयन्ति शालाप्रभावम्.
Optimism is essential to achievement and it is also the foundation of courage and progress. Now I request such an optimistic person, our beloved Dean Madam, Dr. G. Jyoti, Dean of Life Sciences, to brief about our college.
A very pleasant afternoon to all the delegates who have joined in our two-day international webinar on current scenario in therapeutics, prospects and challenges organized by the Departments of Life Sciences of Srimad Andavan Arts and Science College. Before going in for the session, let us have a brief outlook about our Temple of Learning. Srimad Andavan Arts and Science College was established in the year 1996 as a self-financing co-educational institution at Tiruchirappalli, Trichy, Tamil Nadu, to offer value-based and quality education to the downtrodden at affordable cost. The college was founded by His Holiness Sri Rangam Srimad Andavan Swamigal and managed by Sri Ranganatha Padaka Vidyalaya Trust. The college offers 15 UG, 15 PG, 10 MPhil and 12 PhD programs in the Faculty of Arts, Science and Humanities. The college has well-experienced, qualified faculty and good infrastructure which gives the student an ambient atmosphere for learning. It has state-of-art laboratories, computer center, e-resource center, and well-stocked library. Being in the Silver Jubilee year, nearly 15,000 students have graduated from our college with a cumulative of 405 university ranks and 43 gold medals. To add to its credit, the college has completed 5 major, 27 minor, and 10 student projects funded by state and central government agencies, and more than 1,200 research publications in peer-reviewed national and international journals. As far as the life sciences departments are concerned, we offer UG, PG, MPhil, and PhD, and various other value-added certificate courses. The college has a separate research center, SRR CARS, encompassing exclusive instrument facility to carry out advanced research in sciences. It also has a mammalian cell culture unit to carry out in vitro study. The center is recognized as CIRO by DSIR, Government of India, New Delhi. The Life Science Department has completed many major minor projects funded by DBT, UGC, and TNSTC. The college also has collaboration with leading industries. Our research focus is on exploring the hidden treasures and to provide scientific and molecular evidences to our traditional system of medicine and agricultural practice. The Life Science Department maintains a herbal garden with nearly 100 species of medicinal plants, terrace garden, mushroom cultivation unit, vermicompost pit, azolla cultivation, apiculture unit, and CPCACA approved animal house. Students are trained in these units to gain entrepreneurial skills. The departments together and individually organizes many seminar and workshops with aim to endow with an insight to the students, researchers, and faculty on recent updates in the field of biological sciences. To gear up and to update ourselves, we are organizing this international webinar with the theme of Current Scenario in Therapeutics, Prospects and its Challenges. Hope this webinar would help the participants to imbibe some knowledge from our resource person. Thank you. I welcome all the delegates to this international webinar. Thank you.
Thank you, ma'am. Now I request Dr. P. Kavita, Head, Department of Microbiology, to deliver the welcome address.
Very pleasant afternoon to everyone present here. On behalf of the Life Science Department, Srimad Andavan Arts and Science College, Trichy, I am honored to welcome you all to today's international webinar on the topic Current Scenario in Therapeutics, Prospects and Challenges. The aim of this webinar is to enhance one's knowledge about biological therapeutics and emphasize the need to understand the various advancements in this field. I welcome our most respected Secretary and Correspondent Amangi V. Balaji, who always acts upon his prime goal to develop our college in all aspects. A warm welcome to our Dynamic Director, Ma'am, Dr. S. Srividya. I'd also like to welcome our beloved Principal, Ma'am, Dr. J. Radhika, who guides and supports in all of our academic activities. Now, I'd like to welcome our speaker for session 1, Dr. B.H. Venmadimaran, Associate Professor, Marine Parasitology and Jellyfish Ecology, Bonio Marine Research Institute, University of Malaysia. We are grateful to you, sir, for taking out time from your busy schedule to impart your knowledge with us. I also welcome all the other speakers of this webinar. I hope the participants are greatly benefited by your words. Our webinar's important group of the audience. I welcome all the heads of departments, participants, and faculty from a plethora of institutions joining us today. I hope you all find the session fruitful and helpful. Once again, welcome you all.
Thank you, ma'am. The greatest leader is not necessarily the one who does the great things. He is the one who motivates the people to do the greatest things. Now I invite such a leader, Dr. S. Srividya, Director, Academic Affairs of our college, to deliver the inaugural address.
Pleasant good afternoon to one and all gathered here. This is Dr. Srividya, who is away from the college but being with you always. It is my pride and privilege to deliver the inaugural address, which is being a very pride to me for our institution. Our institution is one of such rare institutions which are providing leading facilities and leading technologies and innovative technologies in this current scenario. Today, it is said to be the life science era, and it is very essential and highly important to have such a 2-day international webinar throughout the world of the participants. I hope the entire faculty of life sciences have done a wonderful job with regards to that. I wish you all the very best and encourage the delegates, those who are being actively engaged in this 2 days international webinar. I wholeheartedly thank the faculties as well as the resource persons who has spared their valuable time around the world for this listening this wonderful webinar series. Wish you all the very best and have a grand success and bring laurels to the nation as well as to the college. Thank you very much. Thank you.
Thank you so much, ma'am. A true inspirer has the confidence to stand alone, the courage to make tough decisions, and compassion to listen to the needs of others. Now I request such an inspirer, our beloved Principal Madam, Dr. J. Radhika, to felicitate the program.
A very good afternoon to one and all who have joined here for this international webinar. This webinar, the current scenario in therapeutics, prospects and challenges, is the need of the hour today. We never wondered or we never dreamt that we will be in such a pandemic even just a year before. Today, we are in such a situation that we need new drugs to counter the new viruses and bacteria. They are all invisible, but still they create a great impact on our lives. So, we need to strengthen the industry of medicine for which we are living, and we need to get more and more therapeutics from the sources which are available throughout the world. I congratulate the departments of life sciences for choosing this topic because the focus of research of the college also lies in herbal science. Today, we are going to talk about the therapeutics that are going to be available from the herbal resources and other resources so that we counter and we are able to come out of such kind of pandemics throughout and maybe in future also. I wish the department conducts such programs in future also. I wish this program a great success. Thank you.
Thank you so much, ma'am. Now it's time to start the technical session 1. Now I request Dr. V. Reshmi, Assistant Professor, Department of Microbiology, Srimad Andavan Arts and Science College, Trichy, to introduce the chief guest of the technical session 1.
A blissful note to one and all. I am much privileged to introduce our speaker, Dr. B. A. Venmadi Maran, a man of science, Associate Professor, University of Malaysia, Sabah, Malaysia. He is man of impressive educational background. He has done his doctoral degree at Marine Oceanography, Annamalai University, and his postdoc at Hiroshima University. He has also generated a fiscal support of 3 crores through scientific projects, various scientific projects. Also, he has authored 70 articles in reputed journals. He has also published 5 book chapters in reputed international books. And to his credit, he has got many laurels and awards. As time is only restricted, but its credits to the science is enormous. With this brief introduction, now I invite our speaker to take over the dais. Thank you, sir.
Good afternoon to all. Can you hear me? Okay. Before going to talk, just I want to introduce about my institute. This is my institute, that is Borneo Marine Research Institute, University, Malaysia, Sabah. Please watch this video.
The seas surrounding Sabah are known to have the richest marine biodiversity. The biodiversity of coral reefs and marine resources, mangrove and coastal wetlands, and seagrass beds is among the highest in the nation, if not the world. Borneo Marine Research Institute was established in 1995 as a specialized research unit for research and development in marine science and aquaculture. Our motto is conservation and sustainable development of marine resources. We offer postgraduate programs at master's and PhD levels in the fields of aquaculture, marine science, marine biotechnology, fisheries science, and coastal and marine management. We are also involved in producing undergraduates in aquaculture and marine science. Located at the seafront with a jetty and berthing facilities for research boats, our research facilities are well equipped with fish and shellfish farms, laboratories, and an attractive marine aquarium and museum. These are some of the advantages for education, research and outreach, and a coherent functioning of this center of excellence. University Malaysia Sabah is the only university in Malaysia with 2 on-campus aquaculture hatcheries. The Aquarium and Marine Museum is an important component of the institute, which is utilized for biodiversity, education, research, and public outreach. visitors can have a glimpse of what lies beneath the waves, a display of ecosystems as complex communities of organisms and preserved remains of marine flora and fauna. We are equipped with several research boats and diving facilities for ocean studies. The Institute also consists of the Harmful Algal Blooms Studies Research Unit. The focus is on multidisciplinary research with the aim to assist Sabah government in prevention, control and mitigation of harmful algal blooms. The Endangered Marine Species Research Unit focuses on the conservation of a diverse range of endangered marine species such as sharks, rays, marine mammals, turtles and humphead wrasse. For our niche research and development in aquaculture, the fish hatchery is the foundation of BMRI's aquaculture program. It is a place for conditioning broodstock and breeding them in captivity and producing fish seedlings. We have progressed from the first in the world hybrid garupa between the giant garupa and the tiger garupa species in 2006 by closing the cycle in 2016. The shrimp hatchery focuses on the research for commercially important crustacean species such as lobster, giant freshwater prawn, tiger prawn, and mangrove crab. In 2016, our hatchery has achieved a significant milestone in shrimp aquaculture research, where we have successfully bred the mangrove crab of Scylla tranquibirica. The crablets are important for commercial crab farming and restore the natural resources through stock enhancement programs. Rakan Aquaculture is a unique program for knowledge sharing related to aquaculture between the university and industries. We are also committed to serve the community by way of developing human resources and welfare of the coastal community with an ongoing stock enhancement activity program. Through international networking, our institute has jointly established the UMS Kindai University Aquaculture Development Center, both at UMS and Shirahama Fishery Station, Kindai University, Japan. We will continue to forge networking and collaboration with institutions to share interest in our research programs. UMS Borneo Marine Research Institute, committed to sustainable development of marine resources.
So thank you so much. So hope you have watched the video. So that is the University Malaysia Sabah. We have the institute called Borneo Marine Research Institute. It is a center of excellence, and you saw some of the things like hatcheries. We have our own aquaculture facilities. So it is exclusively used for the research purpose and for the education purpose. So a lot of local people, they can able to come and watch watch everything, and then they can understand, learn like that. We are educating all the local people. So now we can go to our topic, our research topic. This is my talk, natural products. It is an effective approach to control fish parasites in aquaculture. So generally, if you say Malaysia, people know about Kuala Lumpur, right? And even today, when I saw, during the introduction, they said it is University Malaysia. It's not that one. University Malaysia, it is in Kuala Lumpur, but we are here in Kota Kinabalu. That is a totally different area. That is East Malaysia. This is West Malaysia. So Kuala Lumpur is in West Malaysia, and we are in Kota Kinabalu, the state called Sabah, and it's a beautiful island called Borneo. Borneo Island. This Borneo Island has three countries, that is Indonesia, Malaysia, and Brunei. So it is island. It is endowed and enriched with plenty of marine living organisms, completely like ocean area, and also having the plants, the herbs, plants, trees, everything would be like a peculiar form. And monkeys, even the animals, they're all very different. And monkeys like Different kind of monkeys you can see, different kind of orangutan you can see. So many things are peculiar. So it's totally different place called East Malaysia, Sabah. My place city is Kota Kinabalu, where we have University Malaysia Sabah. And yes, regarding this aquaculture. So aquaculture, it was established very well nowadays in Asian condition. And the modern technology of this aquaculture, cage aquaculture, it was originated in 1960s in Norway. So Norway, they are the pioneers in the field of aquaculture. They produced, formed a lot of fishes focused on Atlantic salmon and rainbow trout. But in later stage, like 1970s and 80s, all these aquaculture farm completely devastated because of the parasites called sea lice, that is Caligus elongatus and Lepophilus salmonis. So this sea lice created a big problem in aquaculture in Norway, and it's like $1 billion loss. It happened in 1970s and 80s. So at that time, they started to do research how to control the parasites, all these things like that. And the same culture and same problem happened everywhere. And in Asia, you can see the cage aquaculture. So there is a rise in global aquaculture production from 1990 to 2018, like 527% it is an increase. So 90% of the global aquaculture it is produced from Asia. And 122% there is a rise in total food fish consumption from 1990 to 2018. And this aquaculture is having problem of parasites. There is a threat of parasitic infestation and diseases which could impede the development of aquaculture. The major ectoparasites are leeches, monogeneans, caligates. They are affecting different species of fishes such as groupers, snappers, and sea bass. These are the major fishes which are cultured in Southeast Asian countries. These parasites, which constitute an important group of the marine biodiversity. And related to the proportion of animals in the world, the parasites are living. There are relatively few marine studies have been carried out on parasites, and it's highly limited in the world. Most of the phyla, they contain members of the parasites. Some groups are exclusively parasitic forms. So potentially there are at least 28,000 species of helminth parasites are recorded from 45,000 species of the hosts. Still, we have to study more. It can be an underestimate. As a result of the global climate changes, the impact of pathogenic agents and parasites may also change. That is the research we are doing. And life cycles of parasites also changing due to the climatic alteration. It can lead to the problem for the aquaculture. And you can see this is one host fish which is found with number of parasites. One is called Lepophilus elegans, peniculus, they're all copepod parasites, and monogeneans, different kind of monogeneans from the gill region. So this is a cultured fish. From one host, you can collect plenty of parasites. We studied all parasites and their life cycle, how to control. All these things can be studied if you're having a kind of hatchery, if you're having a kind of aquaculture farm, very, very closely related to your research place. You can see these parasites, an amazing parasites, beautiful one. And these are all parasites can be found from the gill region. So these are all ectoparasites. These are all copepod parasitic copepods, and you can see long egg sacs. They are highly embedded on the filaments of gills. So easily when you are opening the operculum of the fish, you can able to see the gills. When you see, you can able to see the parasites, and you can able to locate exactly and clearly. You can take out these parasites using the forceps or scalpel. And this is an important one, that is sea lice, which completely devastated the aquaculture farms in Norway. You can see only the cephalothorax, not complete body. So it has so many, the, the, the parts, the antenna, maxillipede, and leg 1 to leg 3. These are all like, they are playing a major role. So this is a copepod. Generally, you can see the copepods, they're all free living forms. Plankton, we can say. So many people, they study about the plankton. They're all free living copepods. These copepods, they culture and then they are giving us a feed for the fish. That is different one. But this is completely parasitic form. So they're completely modified mainly in the mouth parts, and then all area, everything is modified for its life, that is parasitic life. And you can see this sea lice impact. So you can see brown color, completely all sea lice which are attached on Atlantic salmon fish. and you can see completely like eroding the skin and taken the, I mean, it is eating the blood tissues of the epithelial tissues of the fish, and you can see here the lesions on the body, and which will lead to the death of the fish. And before that, you will get the disease. So many diseases are happening, and like a bacterial viral infection can happen, and it can create a big problem for the aquaculture. If one fish is infected, whole area, the whole aquaculture farm can be infested. So it happened, I mean, in this research, it was carried out in India. So in Chennai, we found this parasite. It's very common in freshwater. This is called orgulus. This is called fish lice, orgulus, which was reported from cobia fish, which was cultured in the pond, and like one fish which was found with 2000 orgulus like that. So which can be controlled only by the mechanical way. Just we have to use the scissors, or just using the hands, we have to remove all the parasites from the body surface of the fish. So it's not easy to do for all fish. When you're culturing all fish, it's very hard to do like that. But still, we don't have an exact mechanism how to control these parasites like that. For the bacteria or viral infection, people, they can able to give a different kind of pesticide. Like many pesticides are used, many chemicals are used, but for the parasites, it's a bit hard, especially for the ecto and endoparasites. And you can see this another parasite, it's a crustacean parasite, isopod. This is called tongue-eating parasite. So this tongue-eating parasite, it will infect and enters into the mouth region, and it will replace the tongue. So this tongue-eating parasite, that is isopod, which will eat the tongue, and then it is replaced. It is acting as a tongue. So as long as fish has the immunity, they can survive. After that, end of the day, they will die. And commonly, it's like a delicious food, like infected isopod infected fish, which is fried one. In Japan, it is a very popular food, and it's expensive. Exactly in the same way, they will take, fry, and give you. It's very delicious and also very expensive. So I mean, like a Japanese, they're very fond of eating different kind of food. This is one among them. And this is a grouper fish. You can see a number of leech parasites. This is a leech. So here I'm going to talk only about the leech parasites. So this becomes a big problem in Southeast Asian countries. Southeast Asian countries, any grouper culture, sea bass culture, snapper culture, aquaculture farm, they are found with these leech parasites. And groupers, which is a very commonly cultured one. Even when you saw the video, we have a grouper culture. We are culturing hybrid grouper, and we are crossing the tiger grouper and giant grouper and produce the hybrid grouper, which is a popular research here. And it is cultured well, and it's very expensive fish and delicious one. So this grouper, which is belonging to the family Serranidae, and the genus is Epinophilus, it has like a 21 species. People are culturing different kind of species. And this is the fish farm in our locality, the place called Sabah. This is a state, and my place is called Kota Kinabalu. So here we are doing the culture work. I mean, like our research, our hatchery is also having culture, and this is private farms. Private farms, they are culturing. So these cultured fishes are slowly infested by the leech parasites. You can see here infection on the fin. Slowly it is spreading on the dorsal fin, and you can see on the like a face area, and finally you can see everywhere in the body these leech parasites number is increasing day by day. And you can see one grouper fish is having plenty of number of these leech parasites, which create a big stress for the fish, so which needs to be controlled. That is our main research here. That is what I'm going to talk here. So this leech infestation, which is leading to what's called the decrease of food intake for the fish. Of course, I mean, if it is infested, if it is having any kind of disease, definitely the fish will not be very fond of eating anything, which is leading to the reduced growth. And also we are expecting this leech can act as a vector. It can able to transmit the disease from one host to another host. So that is called vector. We do research on this aspect also. And several countries, Philippines, Japan, Malaysia, Indonesia, Brunei, all reported the infestation of this leech parasite, that is from different host fishes. All are cultured one: Epinophilus, Oreochromis, that is tilapia fish, and latest Calcarifer, that is sea bass, snapper, and hybrid grouper. All are like repeatedly it is infected by these leech parasites. You can watch this video. See, on the fin region, completely infested. So we are bringing to the laboratory and keeping for our research purpose. We are analyzing its weight, how it is, eating, feed intake, everything we are checking in the laboratory. Even eye regions, you can see, eye regions also are highly infested by these leech parasites. So this leech, which is called as Zelanicobdella argomensis, which is pathogenic to fish. So it has elongated and cylindrical body, narrowing at both ends. So this is the oral sucker, and this one is the caudal sucker. So this caudal sucker, it is used for the attachment, and oral sucker, you can see the eyes here, the oral sucker, which will be used for the feeding purpose. It will just go and attach on the caudal sucker will be attached on the body surface of the oral sucker. They can able to suck the blood of the fish and then start to do the reproduction, everything. So it belongs to the family Piscicolidae. Piscicolidae is a family which is having this parasite, number of leech parasites, and this one among them, it's called Zelanicobdella argomensis. And you can see different pictures of these parasites, which is taken from the hybrid grouper, that is tiger grouper and giant grouper hybrid one. So people, they try to use the chemicals for the control of these parasites. That is a question. So large number of chemicals and drugs are used in the aquaculture industry. So these drugs are being used based on the availability only, rather than on a methodical protocol linking, identifying the pathogens and prescribing the appropriate administration. It is not carried out just based on the availability of some chemicals and drugs. People, they try to apply and they try to control the fish parasites. So the most important of these chemicals are the formalin. Easily people, they use the formalin at a minimal level. Of course, they are using in the minimal level like a proper dose level, and sulfonamides, tetracyclines, nitroflurons, even oxytetracycline, chloramphenicol, oxalinic acid, hypochlorite, and polyvinyl pyrrolidine. So many chemicals are used. But of course, when you're using chemicals, it can end up with be harmful one, or at least it can cause the side effects for the host and definitely for the consumers. Because unknowingly, if people, they're applying the formalin or some other these chemicals, drugs in a high dosage, definitely it can cause the problem for the consumers. So these toxic chemicals need to be stopped for the eco-friendly aquaculture. That is our main idea here. So to have an eco-friendly aquaculture, we are talking mostly about eco-friendly aquaculture, sustainable aquaculture, without causing any problem to the environment. We have to maintain everything. That is the main motto of any research now. So even when you are eating the edible vegetables, we are going for the organic vegetables, because to avoid the pesticides, all these things, we are going for the organic vegetables. Likewise, if the fish are cultured without using any kind of chemicals, definitely that is a very good thing for the eco-friendly aquaculture. So we try to do like that. We are creating that kind of atmosphere for the fish. So this, if you apply these toxic chemicals, which will be harmful to the fish, human, and also environment, you can see here, even environment will be completely spoiled due to this usage of chemicals and drugs. So we are looking for the natural product. So natural product, it can be applied as a biocontrol agent. So we can take the natural product from different angle. We are taking from these plants. Sabah is this state. It is endowed with a plenty of medicinal herbal plants. Excellent one. So most of the plants, it is having a lot of medicinal properties. So those properties we are checking, and we are also selecting those plants which can be used to control the parasites, these leech parasites in aquaculture. That is our main research, which is going on in successful way. So because these plants may have the bioactive compounds, so that because of the bioactive compound, which is giving a effect to the infestation of the parasite, and the parasites are removed. So if you do like that, it is non-toxic to the fish and human and environment. So it's better to go for the natural product. So why? So this natural product we can collect from terrestrial and also marine source. Terrestrial, we can see a lot of plants, and marine source, we have a lot of seaweeds, lot of seaweeds we have even we have, today is a hot topic is drugs from the sea. So you can take a lot of drugs which will be very useful for the human beings, and also we can do a lot of research for the fish also, because fish is the cultured fish is found with a lot of problems like viral, bacterial, parasite problem. So those compound can be used to control those problems. So these are having bioactive compounds, which is that's why we are choosing this one, and it can be easily available and also cost effective, less or zero side effects, and which will not be harmful for the human beings. So we can go for the biocontrol mechanism. And we have chosen different kind of plants. Now we have like a 15 or 20 plants. We are analyzing, screening, and going towards a plant which is having a very good effect for the fish parasites. So these are the main plants we are using: Dalinia, Safreticosa, Nephrolepis bicerata. And seaweeds, we are choosing one by one. We have a lot of seaweeds here, a lot of plants, like 10 species of seaweeds we can able to collect here. Now we are checking with this Kappa ficus, alvarezi, and sargassum. They're all very common one, and it is also having very good effect for the disease. So we are choosing these seaweeds to control the parasites. And this one, just for the just for the freeness, just you can see here, this is a jellyfish. This is in Sabah. We have a big library. In library, we are using this jellyfish. Lantern, like lights are used, which will be good for the attractant for the tourist or anyone, even for the children. They love to go to the library and study like that. Even you can do like this in your university. So this is just for fun. I have kept this slide. Okay, so when I'm going for this methodology, how we can use, so biocontrol agent, that is a plant or any marine source we are taking, and we are preparing the solvent and aqueous extraction. We are making different kind of extraction. So which extraction will be useful, which will be active for the control of parasites, that we have to choose later. So we are using different kind of extraction, solvent and aqueous extraction, and we will determine its bioactive compounds using the GCMS and LCMS analysis, and we will do the antiparasite activity. We'll just keep parasites, we'll just keep the parasites, and we will apply this extract. We will see the activity, whether it is working out or not. If it is working out, then we'll go for the next one. So fish trials can be carried out, and we can fractionate the samples. Those fractions we can check which is active for the antiparasitic activity, and then those positive fractions, we can take the pure compounds from those positive fractions. Those pure compounds can be identified using the NMR, and also we can check the antiparasitic activity, and finally we can reveal what is the compound which is found in a particular plant which is active against the parasite of the fish. So this is the main idea of the research. So you can see here we are just collecting the fish and cleaning it, drying it, grinding it, extraction, and then freeze dried. We are freeze dried, and then it will be ready for the application, treatment for the fish hatchery. So once we keep it ready, we are preparing one by one plant, freeze dried everything, and then we will collect the samples and work towards our experiments. You can see here different dosage level are used here. You can see that like a more thick one, and this is like a light transparent one. It shows the dosage level. This is a low dosage, this is a high dosage, so that it needs to be dissolved. The dissolving is the most important point. So the any compound which needs to be dissolved. So the dissolve, when we are checking, so most of the parasites are killed in a short time. So we try to check. So based on the dosage, okay, in this dosage we can able to control the parasite on this plant. So every plant is having different kind of effect at a particular dose. And this is one video you can watch. So we choose this fish trial. This is a fish trial we will do. It will just record the leeches and with fish. So with fish, infested fish will be given. This is an infested fish. It is given with that extract, and which is making to remove the parasites from the body surface of the host fish. And there is no any problem for the host. And this one is formalin we applied, and you can see its activity. When you apply any chemical, its activity is very different. You can see here the behavior. So it gets a lot of stress due to the formalin. So that's why we should not apply the formalin or any chemical like that. And if you're applying any plant product, it will not be having any stress. Just it will take some time. It needs more time. Formalin, it will be very effective and problematic for the fish. But the plants, I mean, it will take more time, but you can get a good result. That is our main idea. And this is one plant. We Dillenia saffruticosa, we powdered it, and using the Soxhlet method, we separated the methanol extract and applied for the antiparasitic activity. And we found like a different group, like control level with formalin we checked, and different dosage, 2.5%, 5%, and 10% of the compound. And effect was like on this plant at 2.5%, that is the lowest dose. It took 41 minutes, but for the 10%, it just took 14 minutes. This is a very good, effect. In 14 minutes, it could completely remove the parasites. So we published this work, and also, we can able to reveal the compound, bioactive compound, which is very important. So when we went to the GCMS analysis, we could able to analyze and give information that phenol, benzyl alcohol, even phytol, these are all antiparasitic compounds. So phytol is a very good, it has a very good antiparasitic property, and dodecanoic acid, benzyl alcohol, phenol, all these compounds are found in this plant. That's why which is very effective against the parasite. And these compounds are completely like chemical structures we have prepared of all these secondary metabolites, and using the LC-QTOF analysis, and which is published in a Fishery Science journal. And we are also trying to apply for the egg, so normally people, they do for the concentrating on the fish parasite. Now the parasite's egg we have taken, and we checked with the extract. You can see here. So inside egg, you can see it's moving. We applied our treatment method. We have given the extract. It is moving, and then finally it will be stopped. So it means it is killed. So the eggs are very powerful. It can able to live without water for 15 days. That's why its number is continuously increasing. It is like a cocoon structure inside the animals. That is the developmental stage of the leech is there. You can see inside the developmental stage of the leech. So when it is living normally, and we are giving the treatment method like applying the extract, it could completely stop its activity. So this is very important to control the parasite. We study the life cycle of this parasite. So this is another plant that is called Nephrolopis bicerata, which is very common plant here in Sabah, which is having very good property. You can see here at the 10%, this is 10%, it took just 5 minutes, 4.8 minutes to kill all the leeches. And it at like a lowest dose level, it took 25 minutes. That plant, it took 40 minutes. So it took 25 minutes. So we are working more on this plant, all this activity, how it is happening like that. And if you say that if you want to use the formalin in 3.6 minutes, it could kill all the parasites. It is very effective, formalin, but it's very bad for the human health. And yeah, this fish is completely like a fish. They are having a lot of infested leeches. Those fishes are used, and it took just 29 minutes in the 10% level. It took 29 minutes to kill all the leeches from the fish. And through GCMS analysis, we found the bioactive compounds found in this plant. So phenol, butyrolactin, these are all having, you can see the activity, antiparasitic properties. Benzyl alcohol, phenol, 2-methoxy, all these are having antiparasitic properties, and many antioxidant, anti-cancer properties, antimicrobial properties it has. So these compounds are selected, will be chosen further to purify and find out what exactly how we can use that compound. So this is the compound. It was taken from NMR, chlorogenic acid, ivaline, isoveryl, hexadecanoic acid, methyl ester, and phytol. These are the major vital compounds which is acting as antiparasites. So antiparasitic compounds. So the phytochemical composition study of this on these plants studying is very important. So these phytochemical analysis which indicates the metabolites which are terpenoids, steroids, flavonoids, and phenolic compounds. So this study clearly indicates that natural plant fractions are a vital source of the bioactive compounds with a lot of antiparasitic properties. So we need to do more intensive research on the purification and isolation of the bioactive compounds for the antiparasitic activity. And application is very important. So we are applying for the prevention and treatment of groupers infested with marine leeches. Now we are applying in a like a mass scale. We have to go for the mass scale analysis, how we can able to control or not, like that is the major one now. And also we are characterizing and identifying various bioactive compounds with antiparasitic potentials. We can separate that compound, and that compound alone we can use for the control of these parasites, and which gives a very good impact on the society. It improves the safety of the seafood for human consumption. That is the major thing we are trying to do. And also which increases the food security and production. And also the most important thing is cost effective. So in aquaculture, the cost effectiveness is the most important thing. So if it is whatever we prepare, the compound, everything, if it is very expensive, then farmers may not use it easily. So the farmers maybe should able to buy it easily in a very cheap cost. That is why we are going for the natural methods like normal plants, everything. So cost effective and natural source for the treatment of the infested fish, which is important one. And I can conclude that it's a good source of the bioactive compounds of various natures with antiparasitic properties. Most of the natural products are having like this. And they are showing a strong antiparasitic activity with 100% mortality against the parasite. And the isolation of the pure compound, which is going to be the major one and the tricky one to understand those pure compound, which can be used to control the parasites. And we are going for the like a patent application, everything which needs to be done. So, I have to acknowledge the principal, the dean, and organizers of the Srimad Andavan Arts and Science College for inviting me to talk about in this topic. And also, I should thank Dr. Vidya. She's helping me to connect you in all the like research and these things. It's very interesting. And I have to thank my lab members and all my hatchery staff and my directors. And I have a lot of funds to work on this kind of the project, which is going very well. And this is our research group. My students, they're working here, and I'm also simultaneously I'm working on the jellyfish. So that research also is going on here, and we are publishing this paper. So this is a recent publication in Nature journal, that is scientific reports. It's a Nature group. So antiparasitic potential of this Nephrolepis bicerata methanol, and which is highly interesting one. Many people appreciated this research, and we are going for the patent application, everything. And this is published in Aquaculture Research. This is in Molecules. We reveal the compounds through this GCMS analysis, and we got the gold medal for this research in our university. The novel and effective approach to control the fish parasites in aquaculture. So which is highly appreciable by the fish farmers here. Thank you very much for your patience. If you have any questions, you can ask me. Thank you. Yes, if you have any questions, you can ask me.
Sir, we have certain questions like, so you have done an enormous work on this marine species. How about the parasitology of this freshwater thing?
Hello, can you hear me? Hello, can you hear me?
Am I audible? Yeah, yes, sir.
Yeah, okay. Yeah, so that's what I said. In India, like a lot of freshwater fish culture is going on. So people, they try to apply a kind of chemicals in a very normal level, and they are trying to control in the pond. In pond culture, a lot of this Oreochromis niloticus, Mozambicus, the culture is going on. People, they try to do only chemical application. No one is doing the natural method to control the parasites. And also, like I said, like a mechanical, like a hand by hand removal, just by using the scrappers, they remove the parasite, something like that. But we cannot control the endoparasites. So in India or in many countries, those who are doing pond culture in open form, they are trying to do like chemical applications. There is no much work on natural method. So we have to try with a lot of pond.
So in that case, if you're going for chemical applications, how about the application of alkaloids as you mentioned in your talk, sir? Because this alkaloids is it is reported as toxic agents. When we apply to fish, if it is not toxic to fish, when it is consumed by humans, whether it will cause some harmful effects or it is not like that, it is rather reported.
Yeah, that's what I'm telling. When we are doing this chemical application to really problematic one, but they are going in a very low level of dose, very low dose. So that is a level they are setting. That is a standard one. Otherwise, they can never remove from the freshwater fish. So if anything's like a over level, if it is crossing a particular standard, definitely it will affect the human beings. That's why we should go for the like a natural method without using any chemicals to control.
How about the biocontrol agents? It is actually the source from plants and seaweeds. How far it is being tested, and how about the success rate of using those seaweed extracts and everything?
Yeah, good question. It's a great question. And that's what I'm doing. Many people, many research is going on everywhere. People, they try to use the neem plant. Even I use the neem plant, which is giving a very good effect. And mint is used. Even garlic. Garlics is many. When I go to the fish farm, they are using the garlics. They say we have some parasites, so we use the garlic and it is removed. I don't know how they do like that. So this kind of things, it is going on everywhere in the fish farm. So farmers, according to their richness, like a level of research or level of what they are culturing, they are trying to use a natural method. But when we use like a seaweed, all these things, we don't have much research on this. So without a proper research and confirmation, you cannot use those seaweeds easily in a big level of the fish farm. So if it should not be harmful to the fish, of course it will not be harmful, but it should give a effect. It should kill the parasite. So all this, whatever I'm talking, it is in the like a research level only. It is not highly used for the fish farmers. In a, like a large scale, it is not used. So we have to develop like this to control the chemicals. That's what I'm talking.
So thank you, sir. And there are many questions from audience. So because your topic is so interesting and you have contributed much in your field. So one more, I think this might be the last question, sir, from our side. So does the seagrass has the same effect as the anti-parasite has the seaweeds.
Yeah. Seaweeds are
How
having
about
effect.
using the
Yes.
freshwater sources also comparative?
Yeah, that freshwater sources means I said like plants, that is a freshwater
Yes,
terrestrial plants we are using, which is having a good effect, and seaweed is a very good effect.
how about the
We have,
sea?
yeah, seaweed is giving a very good effect. Even seaweeds that extracts are used to
Yes.
kill the pathogen of the fish culture. So when fish culture, not only parasite, bacteria and virus is the major problem. So this bacterial pathogen, people they try to use the seaweed extract to control it. And also, the harmful algal bloom, that is protozoan, that is harmful algal bloom will be blooming in the sea. So to control that harmful algal bloom, seaweed is used. So seaweed is the major and interesting medicinal organism where in future most of the people or most of the like people they will try to eat the seaweed, and which is very good antioxidant and so many properties it has. So definitely seaweed is a worthy organism to check for everything, even for the human being disease it can be used.
Yeah, yes, sir. Thank you so much. I think exactly you have pointed out the need of the hour because we are in urge for therapeutics, and you have also emphasized the importance of seaweeds in all aspects to be used as therapeutics. I hope it is really interesting, and we also enjoyed your session. It was very informative, and your contribution and your expertise in the field is enormous. And I hope you have been an inspiration for the student group. So I hope many students will be doing some kind of research in this aspect in the future. Thank you, sir. Thank you for your informative session, sir.
Thank you very much. Yeah.
Yeah, sir. Thank you, participants. We are in the end of the technical session one, and we are having a short break, and we will be back by 4 o'clock for the technical session two. Good evening to one and all. I welcome you all for the technical session 2 of two-day international webinar on current scenario and therapeutics, prospects and challenges. To begin the session, I invite Dr. C. Janani, Assistant Professor, Department of Biotechnology, to introduce the speaker.
Good evening, everyone. I welcome you all for the first day, second session of the webinar on current scenario in therapeutics prospect and challenges. It gives me an immense pleasure to introduce the speaker of this session, Dr. Gokul Shankar, Senior Associate Professor, Microbiology Unit, Deputy Dean, Students and Alumni, AIMS University, Kedah, Malaysia. He started his collegiate studies in 1994 in the field of zoology, MS University, Tirunelveli, and completed his MSc in 1996 and PhD in 2007 in the field of microbiology at MS University Tirunelveli. He started his career as a lecturer in microbiology at Chattamada Karayalar College of Arts and Science, Tenkasi in the year of 1996, followed as an assistant microbiologist, lecturer in microbiology, head of the department, microbiology executive, and research scientist in various reputed institutions at Chennai and Bangalore from the year of 1991 to 2009. He has teaching experience of 17 years in various colleges and institutes. He completed PhD 2 students under his guidances, and he has published totally 6 books, 50 research papers in reputed national and international journals with a Google citation of 579 and H index of 9, item index is 9. He also published various general and scientific articles. Professionally, he is a member of Association of Microbiologists in India and Microbiology Society of India. He acts as a member of various professional bodies like Food Scientists and Technologists, Society of Indian Human and Animal Mycologists, Podigai, Enora Innovators Club, Tenkari, Blue Cross Society, Asia Pacific Institute of Food Professionals and Microbiologists Society in India. He is an editorial board member and reviewer in various national and international journals. He organized various national and international symposium, conference, seminars, workshop in his career. He also acted as a member in the AIMS University in the year of 2018 and 2019. He received many awards and he received FRS grant in Malaysia. He have received Young Scientist Award at University of Columbia, Sri Lanka in the year of 2011. SAB Award of Excellence in Food Safety in 2012. Alumni Achiever Award in 2015, Award of Excellence in Food Microbiologist in the year of 2016, and Best Teacher Award in the year of 2019. He had vast industrial experience in quality assurance activity, and he have 5 years of experience in conducting organizing the clinical trials. Today, we are privileged to have you, sir, and I take this opportunity for inviting you on the behalf of our Faculty of Life Sciences team at Andavan Arts and Science College, Trichy. I kindly request you to share your knowledge, sir. Thank you, sir. This session for you.
Okay, I'm just sharing my slides. Is my slide visible? Is my slides visible? Okay. Good evening, Trichy and the rest of the world. It's a great feel to say the word Trichy itself, because I did my schooling for 2 years in St. John's Vestry, and it's always a nice feel to think of Trichy. Trichy is also the greatest abode of Lord Vishnu, the first of the 108 Dividesans. Okay. And in the concept of Hinduism, like it is believed that the word Om is the word which is responsible for the origin of all cosmos. Okay, but my teacher used to say, like even in the scientific world today, the word Om actually it rules the world. You know how? Genome, metabolome, proteome, biome, microbiome, and all ends with Om. Okay, so that's just to start the session, and thank you to the senior management of Srimad Andavan College of Arts and Science and the organizers for this opportunity to talk to you today. In fact, today's talk is on an important topic to discuss the importance of fungal infections which you encounter on your everyday life, okay, and its significance in the newer era, in the meaning like in the era of COVID. Okay, so do you all remember the ship? And okay, so I think, but usually in a webinar, like we just talk and then we assume that you all nod your heads because I cannot see you. But if it's in a real face-to-face session, I can see your body language too. So I assume that you recognize this ship, Titanic. But okay, something interesting about this movie Titanic. Okay, like you can see the Jack and Rose here. Okay, so what is interesting about this movie is like this is the only movie which was nominated for 14 nominations in the Academy Awards, and then it got 11 Academy Awards. The other two movies which were to win the 11 Academy Awards were The Lord of the Rings and Ben-Hur. Okay, so what is that today with this talk? Okay, you know what happened to this ship? Okay, it hit an iceberg, so and it sank. So whenever I go to my class and ask my students what's the specialty of an iceberg, so they will say like, and you will get a lot of funny answers, including like iceberg is made of ice, and then they will say like it's huge, it floats on water, and so many other answers. But suddenly someone will come out with a spark and say like what you see is just a small piece of it. Okay, means like the visible part is just the tip of it, and you have a huge mass which is underneath. Okay, so what I'm talking about today is not the tip of the iceberg that hit the Titanic, but the tip of the medical mycology iceberg. So what I mean by this is like, you know, like in microbiology, the microbiologists study more of bacteria, more of viruses, more of parasites, but what is studied very, very less is the fungi. But this is the era of immunosuppression and immunocompromisation. So if there is no proper management of fungal infections, okay, so what you get as infections is just the tip of the iceberg, but what is below hidden is a major chunk. So means like if there is no proper management, the fungal infections can surpass the bacterial and viral infections in future. Right. So these are some of the reports, like in different papers and journal articles, which says like the alarming rise of the fungal infections. Okay, so it says think mycology, don't ignore mycology. And then the coining of the term, the tip of the iceberg, is not mine, like it's by a famous mycologist, ILO. And so stop neglecting fungi. Okay, I mean the fungal infections, fungal diseases. Right. So and even if you see like among the mycologists, like a lot of people study the plant fungi, not the human or the fungi that infects the animals. Right. So what is that importance of these fungi is like these fungi, a lot of them, they can cause opportunistic mycosis. Mycosis is a term which means like fungal infections. So when I say it's opportunistic mycosis, these fungi wait for an opportunity to strike. And what is the opportunity? When a person is immunosuppressed or immunocompromised, okay, that can be because of various reasons. If the immunity is down, then the fungi finds an opportunity to strike. So what these fungi do is like these, most of these fungi which cause these opportunistic infections are low virulent fungi. They are there in the environment and they wait for an opportunity. So when I say where they wait for an opportunity, I'm a Marvel fan because of my kids. They love Marvel characters. So I always compare this opportunism with Thanos. Thanos was an opportunist. Why? Because he waited for an era to collect the Infinity Stones, an era which was free from all these people, Ancient One, Ego, and Odin. Odin is the father of Thor. Okay, so all these people were capable of stopping Thanos from getting these Infinity Stones. So he waited for a period when all these people were not there, and he started collecting the stones. So that's the opportunistic nature of Thanos. Actually, you can, the other example can be like Thanos waiting for an opportunity, like even he was willing to sacrifice his own foster daughter to get the soul stone. So that's opportunism. Okay. Why this opportunistic fungi are important is like, see, in a normal host, which means like in a healthy person, okay, there are only around 50-60 fungal pathogens which can cause infection, be it a systemic infection, which is deep inside the organ systems, or if it is a superficial infection on the skin, or if it is a subcutaneous infection. There are only 60-70 pathogens. But if you count on the opportunistic fungi, okay, which means like any fungi which is there in the world, okay, which can affect an immunocompromised person. They are put around 300 species, but that's not a limited number. It's a growing number, because any day you will find a new fungi which is not reported before, can cause an infection on a human being. Just by a chance encounter on a human being, it can cause an infection. Right. So as I told you, where do these fungi come from? The fungi comes from the environment. They are basically saprophytic. Saprophytic meaning they live in the dead and decaying organic matter in the environment. So these saprophytic fungi, they are there everywhere. Okay, in the air we breathe. Okay, in the room we live. Okay, everywhere there are fungal spores. Okay, but they don't cause infection. Okay, when your host when the host is healthy, which means your immune system is strong, they don't cause an infection. But when you are immunosuppressed or immunocompromised, then the saprophytic cause infection. Saprophytic fungi cause infection. The other source of these fungi are the endogenous fungi, which are normally in your body. They are called the commensal organisms. Okay, example is Candida. Okay, and they are just commensals in your body, but they wait for an opportunity and strike at the opportunity, okay, to cause life-threatening infections. Okay, so talking of the opportunistic fungi, like if I ask you, say any fungi, I told you any fungi can cause infection. So it's just an exaggerated statement when I say any fungi starting from alphabet A to Z can cause infection. Something like I just related to the Marvel characters, okay, A to Z. So a Marvel fan, if you ask, like, you give me an alphabet for each of this, he will say A for Ant-Man, B for Batman, C for Captain America, and so on. Okay, it depends on the interest. Okay, you ask you ask Vaishnavi to say like give the names of Lord Vishnu in alphabetical orders. He will say A for Aniruddha, B for Bhavo, and so on. Like, it depends on the interest. So like if you ask me again, like as a mycologist, I will give you Aspergillus, A for Aspergillus, B for Bipolaris, C for Cladosporium, and so on. But don't ask me for every alphabet. There may not be for some alphabets. That's fine. Right. Now, there is an upward trend in the opportunistic mycosis, which I said, because of the immunosuppression and immunocompromisation. But it is also because there is an increased clinical awareness. Okay, there is improved laboratory diagnostic techniques and diagnostic tools. And also because the most important factor is like there is an increase in the susceptible hosts. Okay, so when I say susceptible hosts, who are the susceptible hosts? Okay, the hosts means like all those people who have immunosuppression and immunocompromisation. But what are the predisposing factors which make a person susceptible? We will look at that a little later. And also all those newer diagnostic and therapeutic procedures which are mostly invasive. those days, like if a person has got an ulcer in the stomach or an infection because of Helicobacter pylori, like it may be more a clinical diagnosis. Now you go to the doctor, you want to confirm, then he puts an endoscope to see, because the procedures for diagnosis have also become more invasive. So that's also provides an opportunity for the opportunistic mycotic fungi to infect those people. Okay, so what are the things, the predisposing host factors which I mentioned, which can weaken an immune system, is like you have certain inherited immunodeficiency diseases, something like neutropenia, where the neutrophil count is less. Okay, or it can be because of some metabolic diseases. It can be diseases like diabetes. It can be life-threatening or debilitating diseases like cancer, or the treatment which is used for these diseases, something like the drugs that suppress the immune system. Say, for example, in chemotherapy, like the drugs are suppressing the immune system. Same way, like when you are in a prolonged treatment for with corticosteroids, okay, any sort of steroid treatment, then they are more prone for fungal infection. Besides that, the other debilitating diseases or immunodeficiency diseases like HIV, and even in the present scenario, COVID-19. Okay, so which can lower the immune system, can make a person susceptible for these infections. Okay, so usually we say like the beauty is skin deep, but the beauty of the skin can be spoiled by these fungi. So I just want to give an overview of some of the common fungal infections which you encounter on an everyday basis before we go into the current scenario. Okay, so some of these fungi, what is most common is the superficial mycotic fungi, okay, which cause infections on the skin, hair, and the nails. That's the most common one. Okay, and you have a lot of fungi causing these infections. So I just want to discuss on two important things which are very common. One is Pityriasis versicolor, which is caused by the organism Malassezia species. Malassezia is a lipophilic fungus. Okay, and then this is the same organism that causes dandruff too. Okay, and then the other group is the Dermatophytes. Okay, which causes the disease Dermatophytosis. So we'll talk on that. Okay, this is the other the superficial infection, which is called the Piedra, which is this. For the first picture is the black Piedra, the other one is the white Piedra, where it forms a sort of nodules in the hair. Okay, so when I say nodules, it's something like your root nodules. You would have studied about the root nodules in the leguminous plants, like so, like that. It forms the nodules on the hair. So these nodules can make the hair brittle, and then it can break. So these are some of the infections that occur in the hair, right? Before I go into this, like I talk about Malassezia, let me start with most of you will be master fans here, so let me start with a Kuti story. Okay, so imagine if I have a daughter, I would name her as Malassezia. Malassezia is a name as of a fungi. So so I just that's a nice name. So I thought I will call my girl as Malassezia. Okay, so it's in the honor of the East. Okay, I told you this is a lipophilic fungus, meaning lipid-dependent fungus. You have to provide extra lipid for its growth. So that is where this becomes important, because in areas which is rich in sebum, this organism grows. Okay, so which are the areas which is rich in sebum? Okay, your upper back, your shoulders, okay, your upper arms, okay, and also your scalp. Okay, that's why you see the organism like this is the organism Malassezia, which causes dandruff. That's why it's common in the scalp. Okay, so what does this organism do? Like I told you, it causes a condition called Pityriasis versicolor. Pityriasis versicolor is nothing but Temal. Okay, you know, like Temal. I'll show you the picture a little later. Okay, so what happens is like this fungus actually utilizes the lipids or the fats present there. It breaks them into fatty acids, and they damage the melanocytes. Because of the damage of the melanocytes of the skin, so what you get is a color, a specific color in the skin, which is either hypopigmentation or hyperpigmentation. Sometimes it can be light colors, it can be dark color, depending on the nature of the skin. So people with a lighter skin usually develop a hyperpigmentation, and those with a darker skin develop a hypopigmentation. So most of us in South India, like we will have a hypopigmentation. So it will be white patches on the skin. So that's because of the destruction of the melanocytes there. Right. And no, like I said, this girl is finds it more suitable to live in India and Malaysia, meaning because India and Malaysia is warm and humid. Okay, so warm and humid and hot. I mean, hot and humid. So that's a climate which is more suitable for the fungi. Okay, especially the Malassezia group. Right. So this is the thing which I was mentioning, like. Thamal. Okay, so Pityriasis versicolor is thamal. So you see them prominently in the shoulder region, in the back, in the upper back. Okay, sometimes on the face also you can see that in the neck region. Right. So you see them as discrete hypopigmented patches. Okay, so these are fungal infections. I don't confuse this with vitiligo. Vitiligo is different. That is loss of melanin because of the destruction of melanin. So that's here also there is a destruction of melanin, but this is caused by a fungus. So this condition is reversible. So what you need is an antifungal agent. So sometimes in literature you have seen like there is you call this thamal, no? Like when they say it's called alagu thamal and alukku thamal, depending on the color. So if it is dark, they say alukku thamal. When it is light, they call it alagu thamal, because our people like in South India so fond of fair skin. So whatever is fair, they call it alagu. So that's why it's alagu thamal. But there is nothing to do with alagu or alukku because it's a fungal infection. So I just want to quote. Vairamuthu's line, the Kalithu Themalayum Kadoram Machatayum Parpadepo, as if it's a good thing to have a themal in the neck. Okay, so right. So I was talking about this girl Malassezia again. Her favorite meal is spaghetti and meatballs. Okay, spaghetti is like vermicelli, and meatballs is the soya balls which they use it commonly in the food. Okay, so we say spaghetti and meatball appearance because when you take this fungi and observe it under the microscope, it has got sort of a filaments formation, okay, a hyphal formation, and then it also forms a meatball like structure. That's why I said her favorite meal is this, and she likes cheese. Why cheese? Because I already told you it's a lipophilic yeast, so it needs lipid for growth. So this is what you see it under the microscope. You just do a skin scraping and put it on potassium hydroxide and see it under the microscope, you see this appearance. So hope you are listening, bro. Right. And dandruff is a very embarrassing condition, Nanba. So why I say this is like, if I ask you in the whole group, like how many of you have dandruff? Okay, if it's a big hall where we normally have a seminar, like none of you will raise your hand because it's a sort of an embarrassment. There is no need to be embarrassed. It's just a fungal infection. If anybody asks you if you have a any other bacterial infection, okay, you will say yes, I had this sore throat, I had this boil, and so on. So something like that. This is just a fungal infection. There is no need to be embarrassed about it. So I was saying, like this girl, I told you again, Malassezia is an organism which also causes dandruff. So I just symbolize that the girl has got a long hair and then she has dandruff. So what I need to gift her whenever she gets her birthday is gift her with shampoos. Okay, but make sure that shampoos have an element which is an antifungal element, whether it's a zinc pyrethron. Usually it's put it as a ZPTO on the packs. Okay, or it should have some compound like selenium or climbasol and so on. So like then you see any of your shampoos, anti-dandruff shampoos which you use, which you find in the market, like Clinic All Plus, Head and Shoulders, or Nizzerol for that matter, you will have any of these antifungal agents. And this is something which you need to understand. Okay, if your advertisements, shampoo advertisements say you that we can cure you of dandruff, then it is a big lie. Okay, so that's one of our report where we said that was published in Deccan Chronicle. So what we say is like, this is a lie. Why? Because dandruff cannot be cured, but can be effectively managed. Why? I told you this Malassezia is a commensal flora. Okay, it is a normal flora of the skin. So you cannot eradicate the normal flora, but you can keep it under check. How? You can keep it under check. If you have dandruff, then you have to use your anti-dandruff shampoo twice a week, or depending on your severity, twice or thrice a week. But if you leave your anti-dandruff, say in the sense like you get rid of dandruff and then you stop using your anti-dandruff shampoo for 4 to 5 weeks, then what happens? You get all your flakes again. That's because the organism will start growing again. As I told you, it's a commensal flora. But what is so interesting about this organism is like it is there present in the scalp of every individual. I told you it's a commensal flora. But why it causes dandruff only in certain people is $1 million question. Okay, so there can be several factors which are actually host factors, not associated with the fungi. It's host factors. Okay, more of sebum production. Okay, or other conditions like which can favor the growth of fungi. Right, so this is Malassezia again. Okay, the second major group of organisms like which are most common in causing skin infections are the dermatophytes, and the infections caused by them is dermatophytosis. So they basically are very fond of keratin. Okay, keratin is very rich in the nails, the skin, and the hair. So these fungi, they basically cause infection of the hair, the nails, okay, and the skin. And what is interesting about these dermatophytes is like these dermatophytes can exist in soil. So when they are living in soil, they are called the zoophilic. They can exist in the animals. They can live on animals, so animal fur. So they are called zoophilic. And then some of them are anthropophilic, which are living only on human hosts. Okay, so no other organism. Okay, no other organism, the fungal organism, like it has got such a wide, interesting nature that it can be in all the three places. It can be in the soil, it can be in the animal, it can be in the human host. Okay, but some of these fungi, some of these dermatophytes have totally evolved as human parasites. Okay, so they don't have a survival mechanism in the soil. Okay, some of these species have totally evolved, okay, only to be as obligate human parasites. Okay, so these are the three major genus under which we have the dermatophytes: Microsporum species, Trichophyton species, and then Epidermophyton flocosum. Why Epidermophyton alone with the species name is like this species Epidermophyton has only one species, so Epidermophyton flocosum. The other species Microsporum and Trichophyton have lots of species. As I told you, it can infect dogs, cats, people. Okay, it can infect cattle. Okay, so now this is something interesting. I told you that these fungi can live in the soil, live in the animals, and then also cause human infection. Okay, but whenever there are some organisms which live only on the human host, when they cause infections, the infections are not severe. Okay, but whenever the organisms which live in the soil or which live on the animals, they cause fungal infections. Okay, skin infections. These infections are very, very severe. Okay, so that's something for you to think through. Okay, if you can think through, like it's good, like, or we will discuss later during the Q&A. Okay, dermatophytes is as good as God. It doesn't discriminate anybody. So it treats everybody in the world equally. So it has got a geographical distribution worldwide. It infects all skin types. It infects all the people of the world, whether it is a Caucasian skin or a black skin or a mulatto skin and so on, right? And okay, transmission. I told you these dermatophytes can infect the animals. Say, for example, Microsporum canis is an organism which infects mostly the cats and dogs. And most of these are our pet animals. So, and there is always this tendency that we hug our pet animals. Sometimes some of them so fond of these animals that they take them even to their beds. Okay, they sleep with these animals. Okay, the dogs and cats. Okay, I'm not saying that it's wrong. What I'm saying is, like, if these pets are infected, okay, if they have a dermatophyte infection, then definitely they will pass on this infection to you. You can see that this cat is infected and this dog is infected. Okay. Right. So how do you see these lesions? Or it's like, especially most of the times when the lesion starts, it's a small scaly lesion, localized and round in structure, and then it starts as after some time it becomes itchy. Okay, pruritic. So that is the reason, like you will start concentrating on this, or you will get you start scratching it, and sometimes you ignore. When the itch becomes too much, then you go to the doctor. Okay, and why it is? Because it is favorable, like the hot and humid climate, the climate of India and Malaysia, it's or most of the Asian countries, it's very, very suitable for this dermatophyte infection. Okay, and there can be other predisposing factors. People with diabetes, steroid, who are under steroid therapy, they can be predisposed to dermatophyte infection, but not necessary that only people with diabetes get. Anybody who's a healthy host can also get these dermatophyte infection. Okay, so depending on where these infections occur, okay, these dermatophyte infections are classified into different types: Tinea capitis, corporus, cruris, anguem, barbae, and pedis. So we'll see some pictures of those infections so that you'll understand that much better. So this is Tinea corporus. It can occur anywhere in the body. Okay, Tinea infections are also called as, the dermatophyte infections are also called as ringworm infections. Okay, ringworms because it produces a ring-like structure. Earlier they were thinking that these were caused by parasites. That's why it's called ringworm or tinea. Tinea, actually, it's a word it's used to refer parasites. So they were thinking that these were caused by worms, but later after we studied the fungi, we understood that these are nothing to do with the parasites. They are caused by fungi. But because of the popularity of the term, even the medical books today, they use the word tinea or ringworm, okay, in it. So it's a misnomer. It's not a proper term to use, but still it's popularly used, right? So these are in this is tinea corporus, which is a lesion on the smooth skin. Okay, so you can see these lesions, circular lesions all over the body. Okay, and then it becomes pruritic. And if you see, the nature of these lesion is like you will have a raised margin, okay, on the edges. Okay, the edges will have a raised margin. Okay, this is Tinea pedis. Tinea pedis is like actually the tinea infections or dermatophyte infections. You call it in Tamil as padar thamarai, padai, and all that. So Tinea pedis is pada padai. Pada padai means like the fungal infection of the feet. So it's also popularly called as athlete's foot, because athlete's foot those days was common in athletes because they always use the shoes. Okay, so but not necessarily only athletes should get it. Everybody can get it. People who these days, so who are sophisticated, we have enclosed footwear or shoes, and we live in a hot environment. Okay, a hot and humid environment. Okay, then it's not suitable, so you develop the fungal infections, right? You always tell this is a famous thing, no, right? You always say like you need to be going to other man's shoe, or just step into other man's shoe just to know that you don't misjudge them. So you always say like never judge a person, like you should be in their place to judge them. So before you switch to their shoes, make sure that you don't have he doesn't have an athlete's foot. So otherwise you are going to get the fungal infection, right? So this is Tinea pedis or athlete's foot. And you can see that they sometimes they call this as setupun. Setupun meaning like, but nothing to do with the ser. Okay, but the only thing is like I told you, the hot and humid nature. But people who have an occupational hazard of always like setupun, because farmers and all, they are always their feet are always wet. Okay, and then it's exposed to environment. They don't wear a footwear. So then this possibility of getting the tinea pedis is more common. So you'll see that in between the toes, you'll get these in the intertrigeminal region. You can see these lesions. Okay, and the tinea unguem is the nail infection, also called as nagasutti. Okay, and the popular Tamil movies those days and all, if somebody has a Nagachutti, they will put a lime over it and come like this. Okay, like Krishna coming with the chakra and all. But imagine like, but doesn't help. You need a antifungal treatment for that, because the nail infections are very severe. Like if you don't treat them on time, like all the nails are going to get discolored and going to change a different color. So you don't need a nail polish. The color itself is so eminent. You get the brown color, yellow color, and blue color. All this, you don't require any nail polish after that. So, but it's a fungal infection, so you need to address it. And then when it is a fungal infection of the nail, it is very, very difficult to treat. So most of the times, if you use a topical cream, okay, or as a lotion or a gel over the infected area, for if it is a skin or if it is your on anywhere on your body, okay, on your face, okay, it may respond better for a topical cream, but it may not respond properly for a nail infection, because the fungus actually goes and lives deep in the nail bed. So you need a oral therapy, okay, oral antifungal therapy for such conditions. Okay, that's what I discussed, that nail bed infections are difficult to cure. Okay, so this is an interesting case where women was treated for dermatophyte infection. Okay, it was an intractable case. Intractable means like they were treating, but it was not responding for a lot of time. So that's case. Then she had tinea corporis on the body, like, and then like it was recurring. Then it was not responding to the antifungal treatment. And then when they noticed, like, she also had a nail infection, so you can see the whole finger is swollen. Okay, so she had a nail infection and she was transmitting. Even like if these lesions are getting cured by scratching, she will transfer it to anywhere in the body. So finally, they had to give a treatment of oral antifungals. Okay, so they initially tried with all the topical cortisone creams and all, did not work, and then finally had to take a 30 days course on oral itraconazole to have it cured. Okay, tinea captis is the infection of the scalp. This is more common, like usually like in children. Motta potadukku appuram pathina, then you will get these lesions, because that is if the blade which is used or this thing is not properly sterilized and all, it can transmit from one person to the other person, so it can cause tinea captis. Okay, otherwise also, like if the fungal infection can occur. So this is again tinea captis. Okay, this is Jock's itch or tinea cruris, which occurs in the crural region. So it's in the occluded region, and as basically you need this is one condition. People, if they get the infection also, they do not disclose, common in men and women, and that is because of a lot of sweating in the area. You wear tight occluded cloths, so the chances of getting infection is more. So next, like if you or any of your friends or anybody experiences this sort of discomfort or itching feeling, like, then better to consult a physician or a dermatologist and get it cured. No need to be embarrassed about it, because it's just a fungal infection. Okay, so this is a severe tinea cruris because a person did not go to a doctor for a long time. Right. So we always make fun of people like who put both their hands inside the pocket, and we always ask whether it is because of style, because a lot of people put their hands inside, whether it is because of style or because of Jock's itch. So make sure that we don't get Jock's itch, because. And then the other one is like the infection of the bearded area. So it's called tinea barbae. It's again a dermatophyte infection. Okay, so all these dermatophytes, I told you, like they belong to these three genera: Trichophyton, Epidermophyton, and Microsporum species. Okay, so they can infect the hair, skin, and nails, and there are individual variation between the different genus that they infect different areas of the keratinized tissue. Okay, now talking about the therapy, there are a lot of drugs, antifungal drugs, which are available, but the identification of a fungal, because I told you it can be caused by 3 different genus and a lot of species are there, and finding out which species cause the infection doesn't help in the treatment, because like the therapy is the same, and then irrespective of whether it is trichophyton or microsporum, then the treatment is the same, so it responds to the treatment. So most of the time they don't go in for the culture of the fungi, but the culture of fungi will be very important to identify the source of infection as well. Say, for example, I told you there are certain fungi like Microsporum canis, which are zoophilic, which can be in the cats or dogs. So by chance, when you do a fungal culture of your dermatophyte lesion and you find that Microsporum canis growing there, then you know like the source of infection is the pet animal in your house. Okay, so that also, otherwise you will be getting recurrent infection in spite of your treatment. Again, your pet is going to infect you, or you are going to infect the pet. It is going to be a continuous chain of events. So that's where the identification of the fungi comes into importance, right? So what do you do? I just want to discuss one thing which is very important for all of you to know, like, is because the dermatophyte is very common. I told you 90% of the people in the world get any form, at least one form of dermatophyte infection in their lifetime. So we need to be aware. Most of the time we are not aware. Sometimes some of these lesions are self-resolving. They go on their own without any treatment as well. So there is a rule which is called the 2-2-2 rule. With what is this 2-2-2 rule? Is like you use any antifungal cream for the dermatophyte infection. You use it for 2 weeks. You have to use it 2 times in a day. Okay, and then you have to use it 2 centimeters beyond the margin of the lesion. So when you have a lesion here, okay, 2 centimeters beyond the lesion, because the actively growing fungi will be there in the margins. So you use it 2 centimeters beyond the margins, then the chances of getting cured will be much better, right? And then in a climate which is hot and humid, something like a jeans, a tight fitting jeans, leggings, jeggings, all those are not suitable. Only loose cotton garments are suitable for you. Okay, but if you are in an air conditioned environment, that is different. Whatever you wear doesn't matter because you don't sweat. But if you sweat a lot and then you tend to wear all these tight fitting clothes, then you have the possibility of getting these infections. So the other things, what precautions is like in a climate like that of India and all, like you dry your clothes after washing in the sunlight, right? So that kills most of these dermatophytes. And most of the time, the transmission is through the sharing of things like towels, bed linen. So a family member is infected, a person in the family is infected, there is a chance that everyone in the family can get infected if you share using the towels and other things. Okay, so this is a study from the Rutgers University, US, which says like a lot of people go to the beauty salon, okay, to get your skin more beautiful. But it says 50% of the people who go to the beauty salons end up in getting a fungal infection. Okay, the risk of 50-50. So imagine like you want your skin to be more beautiful and you end up with a fungal infection, right? So other things like wearing a tight wristband, waistband, okay, and then for a climate like India, non-occlusive footwear, like with shoes or open footwear, which will not allow the sweating to happen. Okay, so that's preferable. But if you are a professional, then it requires you to wear shoes. Then you need to take other precautions. Okay. Right. So the other organism which I want to talk about today is like Cryptococcus neoformans. Okay, this is an organism which is common in the it commonly finds its reservoir in the droppings of birds, especially the pigeons. Okay, and these droppings of these birds, you will find it in every place. Like you go to the churches, you go to the mosques, you go to the old buildings, temples, everywhere you get these pigeons and you get the droppings. And definitely all these droppings have these Cryptococcus neoformans. But if you happen to inhale the spores of these Cryptococcus neoformans, provided you're healthy, then you're fine. But if you have any sort of immunosuppression, okay, if your immune system is down for some other reason, for some other condition, then you are susceptible for infection. Okay, there are even reports that Cryptococcus has caused infection on healthy people. Okay, what will happen, like if I get a Cryptococcus infection? Usually it's a lung infection. Sometimes it can self-resolve, but depending on your immune system, if your immune system is weak, it can go and infect your meninges, it can infect your brain, it can infect your central nervous system. Okay, that's a danger. Okay, so this is one of our study done way back a long ago in 2003 and 4, where we demonstrated that the droppings of these birds, like not only the pigeons, a lot of other birds can also host Cryptococcus neoformans. Okay, we demonstrated that these droppings of normal crows, okay, they also have the Cryptococcus neoformans. So, and again, these are some of our published reports which says bird droppings can be fatal, okay, when we are immunocompromised or immunosuppressed. Okay, so Cryptococcus neoformans actually, like it's interesting, like this is a negative staining of Cryptococcus neoformans, like like in the negative staining, what you see is like the background is stained, the cells are not stained. Normally when you do a staining like a gram stain and all that, the cells are colored, the background is not colored. Okay, in a negative staining, the background is colored, the cells are not colored. So you can see in a negative staining that Cryptococcus neoformans is, these cells are capsulated cells. They have a capsule, so they look like a moon with a halo zone. Okay, so normally when you see a moon, okay, a full moon on a dark sky, what does it mean? It means it's a full moon day. Okay, so you see a full moon on a dark sky, then it is a full moon day. But if you see multiple moons, okay, on a dark sky, then probably you are looking into the negative staining of Cryptococcus neoformans. Okay, so this is another fungi, which is an opportunity fungi, which causes debilitating conditions. Okay, whenever it's a immunosuppressed or immunocompromised condition. So especially in HIV people, like this is critical organism which causes infection. Okay, the other most common organism which I want to talk to you about is a Candida. Okay, which is an example of an organism which is a commensal flora or endogenous organism which can convert into, it's an opportunistic pathogen which can convert itself into a pathogen provided a condition is provided. Okay, so there are a lot of other conditions which we already discussed, like prolonged antibiotic therapy, prolonged steroid therapy, and all that. Okay, most of the times you see like Candida causes superficial infections, like the subcutaneous infection, it affects the mucous membranes and all that. But if a person's immune system is compromised, they cause severe systemic infections. Okay, and normal thing is like it can be common in infancy, old age, and pregnancy. That's quite common, because if you're seeing like newborn babies and all, their tongue will be coated white. Okay, so people people who come to give bath and all, all those ayahs and all will say like you have to clean the tongue and all. No, don't do it. Okay, it's just a fungal coat of Candida, which will get resolved because the immunity will be less, so Candida coats on the surface. But if the infection is severe, then you require a therapy. Your pediatrician will recommend that. And okay, so these are the pictures. I'm not going into the details of this, but okay, so one important thing I told you, like this Candida infections, the oral infections caused by Candida, which is called as oral thrush, can be used as a marker for HIV, okay, for AIDS. Okay, if a person is like immunosuppressed and immunocompromised, and you have the oral lesion, like then they say like it can be used as a marker to find out the HIV status or AIDS status of a person. And why Candida is more important is like majority of the women, okay, they experience at least one episode of Candida infection. Okay, they like whenever you get some infection, what you do is like you use a lot of antibiotics. Okay, because of these antibiotics, all your friendly bacteria is wiped off. Okay, and then it because these friendly bacteria are replaced by Candida, then they grow and they cause an infection. So this is very common in the women, like and they can cause vulvovaginal candidiasis. Okay, especially when they are there can be other hormonal factors as well. I told you, like even during pregnancy, the chances of a person getting infected is more. Right. So these are some important members of Candida. Okay, Candida krusei is now renamed. Okay, it has got a different name because it is moved from Candida. Its genus. It's now become Pichia now. That's fine. But the one important addition to this is like Candida auris. Okay, so we'll discuss more about that. That's one potential pathogens which is emerging. Okay, so Candida can also cause infections on the nails. Okay, like how the dermatophytes cause the nagachutti. It can be caused by both Candida species and also the dermatophyte species. Okay, this is what I was telling you about the oral thrush, the white patches on the surface of tongue. This is an indication that the person is immunosuppressed or immunocompromised. Okay, or something is he's in a debilitating disease or in a treatment and all that, or he's in a prolonged treatment and all, then he can show these symptoms of oral thrush. Okay, but don't confuse this with a normal unclean tongue. Huh? Okay, so if your tongue is not clean properly, there'll be white in the morning. Like, so next time you see somebody, like don't ask him, okay, show me your tongue. Okay, it's white in color. Okay, but better you go and test yourself for HIV because you may have oral thrush. Don't conclude that. Unclean tongue will also look white, but this is very discrete white patches which you can see. Okay, prominent. Okay, like this is more like a curd-like plug on the surface of the tongue. Right, and then laboratory diagnosis. I'm not going into these details of laboratory diagnosis. Normally, like what they do, high chroma agar, different colors for different colonies. Okay, this is a very basic thing. The future is going to be the molecular diagnosis. Like one of my PhD student is currently working on the molecular diagnosis of to find a standardized molecular technique for Candida. Right. So I was talking about the Candida auris, and then what is a new global threat? What we have from Candida auris is like all over the world, the Center for Disease Prevention and Control, they have alerted. Like they are worried about Candida auris for the following reasons, because they are resistant, multi-drug resistant. They are resistant to all the antifungal drugs which are already available, which are used for treatment of Candida. Okay, and then the second important factor is like all the labs are not equipped to identify Candida auris. Okay, that's because this is a new fungus. Okay, and it can mimic other species, and then you have to identify only with a molecular technique. And then like I told you, like my PhD student is currently working on this. Okay, so maybe if we get some good results, we will share it in the future. Okay, and the other important thing is like it can cause outbreaks in the healthcare settings. Okay, so if a person is infected and then it can create an outbreak in a healthcare setting, means like it can infect more people in a hospital. So that's are the challenges, because this is multi-drug resistant. Okay, and then difficult to identify, and then it can create havoc by outbreaks in the healthcare settings. Okay, so the other thing is like why we need to worry about Candida auris is like people can carry it on the skin. Okay, just it can be carried on the patient's skin without causing infection also, just spread to others. Okay, just passive carrying. Or this one is like Candida auris, I told you, like it's multi drug resistant, have to several antifungals, but it is also resistant to several of the healthcare disinfectants. So that's also a worrisome factor. The other important factor is like they found that with the advent of the COVID-19 pandemic, they have found that the outbreaks of Candida auris is on a rise. Okay, especially on acute care. Okay, normally Candida auris does not cause infection in acute care. Okay, so usually it will be with a chronic care. Okay, prolonged hospital stay and all, only then people get an infection. But in acute care, it was not common before. But with the advent of COVID, okay, they found that it is it can also cause infection in acute care hospitals. Okay, this is a report which is an important report. Why I want to say is like this describes a case of a person infected with Candida. He was Candida auris, and he was in a hospital for 90 days. Okay, and he died finally, and despite his best treatment. But what they found is like when they tried to clean the room, they were just taking samples from the room. Then they found that all the walls, the bed, the doors, the curtains, the phone, the sink, the whiteboard, the poles, pump, whatever was there in the room, no, everything was infested with Candida. Okay, and also they found that some of these tiles were infected, and then they couldn't do it, so they have to rip off some of these tiles in the ceiling and the floor. Okay, because it has gone deep in them, and then it was infesting there, so they have to rip off the tiles to clear this Candida from that room. So what I'm saying is like this is also a persistent by fungus, persistent and also multiple drug resistant. That's a worrisome aspect. Okay, so this is a global thing to show like the Candida auris reports. Then you can see that like there are several isolated reports, okay, of Candida auris in India as well. Right, and the new challenge. I told you fungal infections. Okay, well, what's a new challenge? Especially that opportunistic fungal infections, which can be life-threatening. Okay, in the COVID era, what is the problem is like most of the symptoms of these fungal infections, which cause respiratory infections. Okay, especially the aspergillosis and invasive candidiasis, all that can be mimicking whatever the symptoms of COVID-19. Okay, which includes fever, cough, shortness of breath, similar. Okay, and then most of the time we do a test, we think it's COVID, and then we may be giving management only for COVID, but that can be a deep fungal infection which is undiagnosed. Okay, so laboratory testing is necessary to determine whether a person is infected with co-infected with fungal infection. It can be COVID-19, can also have a fungal infection at the same time. Why? Because these people who are in the ICU, okay, on all those, they are more vulnerable. Okay, if the fungal infection is not already, because of COVID-19, there is a drop in their immunity. Okay, so if these are not properly diagnosed at the earlier stage, aspergillosis or invasive candidiasis can kill a person. Okay, so again, as I told you, like they are resistant to antifungal treatment, and then whenever there is a severe COVID-19 patient, then fungal infections become resistant for treatment. Okay, and then they also find that several other fungal infections like histoplasmosis, blastomyces. Okay, and then they have a very similar pattern. Okay, of symptoms to that of COVID-19 infections. Okay, so all these respiratory infections, okay, can come very easily, because I told you most of these fungi, all these systemic fungi which cause these infections like histoplasma, blastomyces, and all that, they live in the soil. They are saprophytes in the soil. So if a person inhales it, okay, and then they can get these infections, because if they have already infected with COVID-19, then the possibility of them getting infected is more. Say, for example, histoplasma is there. They find their reservoir in the droppings of bats. Okay, so so if you they go to an adventure sport like caving and all, where there are a lot of bats, then there is a chance that they can get this infection. Okay, so this is another report which is relevant in the current scenario, where from India. So it says like there is an increase in the rise of mucormycosis. Okay, and then this is a eye infection which is caused by mucor, and then like if it is not properly managed, then it can spread to the brain. Okay, so normally if you see, mucor is a bread-eating fungus. Okay, it's a fungus which is normally there in the air. Okay, everywhere. Okay, so you keep the bread for a few days, it spoils the bread. So normal fungus like rhizopus, mucor, penicillium, aspergillus, which is there everywhere in the environment, can cause opportunistic fungal infection. So, but what is the severe about this mucor? Like It's not just bread-eating fungus, it can be flesh-eating mold. Okay, this when it colonizes the sinus cavity, okay, you can see how the severity of the infection. Then they can spread to the brain. They can cause rhinocerebral mucormycosis, and it can be fatal for patients. Okay, so a lot of other risks are there, like other COVID-19 associated fungal infection is pulmonary aspergillosis. As I told you, aspergillus is one common fungi which is abundantly in the environment. So you take any air sample in any of the rooms, any of the indoors, okay, or outdoor, okay, you will definitely have these spores of aspergillus growing. Okay, so that is a major risk factor, okay, when a person is immunocompromised, or if there is a lowered immunity because of infections like COVID. Okay, so mycologists have more fungi to study, more fun also to study during the immunosuppressive or the COVID era. Okay, so what are the scope is like you have to, there are no new drugs, okay. Generally, like they use lipid amphotericin. Okay, amphotericin is side effect because the challenge is like you are dealing with a eukaryotic organism. You have to kill a eukaryote inside a eukaryote. It's different from what you do for a bacteria because it's a prokaryote. So killing a prokaryote is much easier than a eukaryote because fungi or eukaryotes. So addressing them, eukaryote or treating a eukaryotic thing inside the eukaryote, which we are also human beings eukaryotes, so that becomes difficult. So you have the third generation azoles which are used, okay. Then the echinocandins, okay, those are the class of antifungals which actually attack the cell wall of fungi. Okay, so but with this immunocompromisation, what is a aggressive therapy mode will be like there are three approaches which usually people do. One is a prophylactic approach, means like when you start a chemotherapy for a cancer patient, or like a person is immunosuppressed or compromised, that there is a possibility that the person will get a fungal infection. Okay, so if you have a possibility, then you start antifungals as a prophylaxis, okay, before you start your chemotherapy. So that is one approach. Second thing is empirical. When a person is at you start a therapy, a person with risk, which means there is a high fever of unknown origin, okay, and he is not responding to antibacterials, then the second thing what you think is like it can be a fungal infection if his immunity is down and all. Even if you have not tested and found any fungus, then better to start an empirical therapy that it can be a fungal infection. The last one is a preemptive therapy, is like you establish that there is an evidence of fungal infection either by serology, or you isolate the fungus. After that, you start the treatment. So all these three approaches are important: prophylactic, empirical, and preemptive in the era of immunocompromisation. Okay, and the other major challenge, I told you already, the person is immunocompromised and immunosuppressed, then you need to use sort of an antifungal therapy along with the immunotherapy, which means like you have to combine antifungal therapy with interferons, colony stimulating factors, interleukins, and so on. Okay, so and the end game, who won? Who won? It's Thanos or Iron Man. You never know, like, because though Thanos dies, Iron Man also dies. Okay, so end game, who wins is like, but in the when it comes into the immunocompromised host, it is a fungi that always wins. Okay, so that's my favorite slide. I always put it on the class because after I teach something on the fungus, I say like it gives me a chance to mold the young minds. Okay, mold referring to the fungi there. Right. So this is my this is our university, our beautiful campus, Ains University, Malaysia. Okay, so if any of you happen to make a visit to Malaysia, so you are welcome to visit this beautiful campus. You have any feedback on the talk, or if you have any questions which you want to discuss later, you can email me. Thank you.
wonderful lecture. And participants have posted some of the questions, sir.
Okay.
Um, One of the participants have asked how to prevent the hair fall which is caused by dandruff.
Okay, hair fall. There are a lot of other reasons, like say I have a hair fall that is more genetic. Okay, it can also be triggered by hormones, because the, uh, the sex hormones, especially androgens, actually they have testosterone and all have effect on the hair fall. But as you said, like yes, dandruff, like you want to control hair fall which is because of dandruff, then what you need to do is, like as I told you, like for dandruff you have to have an antifungal shampoo, which you have to use it twice a week. Okay, and or twice a week depending on once a week depending on the severity of your infection, and that you have to use it lifelong if you have dandruff. That's the only way. But certain anti-dandruff shampoos, like depending on your skin nature, like it may have uh, a little hypersensitivity reaction also. So you need to check out which one suits you. Okay, and if you still have hair fall, then consult a dermatologist.
Yes, sir. Thank you, sir. And actually, there are so many questions, sir. Participants are overwhelmed on your lecture, sir. So we would like to ask all the questions, sir. And
Okay.
does applying coconut oil helps to reduce the dandruff?
Okay, actually, like whether coconut oil has got a beneficial effect is a question mark, because it's more as a moisturizer. Okay, so it moisturizes, and then it's good for the hair, like which has been traditionally used and believed over centuries. We have established that. But as you said, like it is not that external oil which you apply which causes this organism to grow. There's certain host factors. That's I told you, like because this organism is the commensal flora. Okay, it is there in the scalp of everybody. So even if you don't apply oil, if you are predisposed because of your more sebum secretion, you will get dandruff. So it's nothing to do with the this thing. But there are some studies that they have used that some form of coconut oil along with other antifungal agents, because they use some herbal antifungal agents in them, like it helps to prevent dandruff.
Yes. Thank you, sir. And one another question: how many days does it take to cure tinea pedis infections?
How many days to cure?
Tinea pedis infections.
Okay, tinea pedis. Okay, like usually you will require a treatment of 3 to 4 weeks. Okay, normally, but you have to make sure, like what are the reasons that contribute to that. Okay, tinea pedis, as I told you, like if your moisture in uh, the skin is responsible for the infection. Like a lot of people have a habit of taking a shower, and immediately, because you have to rush to the college or your workplace, you don't wipe your body clean, dry. Okay, so you still have moisture on your body, and then you wear your clothes. Okay, so that moisture actually can promote, yeah, can be a favorable environment for the fungi to grow. So similarly, if you sweat a lot, then preferred, like you should have a open type of slippers, not an occluded shoes. Okay, and then some people, like they anatomically, if you see their the toes, no, they will be touching each other. Some of them are their anatomy is such a way that the toes touch each other, so that it can like rub each other and cause an aberration. So in that aberration, like you have a moisture, then the fungi can come and grow there. So it's better that you need to have an occluded shoe, and then a treatment period like will be 3 to 4 weeks. But if it's a nail infection, then you may recur, other than your topical cream, you may recur your oral therapy also, antifungal therapy. And I would say, like don't always rely on the over the counter products. Don't go, like you don't be a doctor yourself. Like go and discuss with a dermatologist or a physician and get a proper medication to get it treated.
Yes, sir. Thank you, sir. And if the nails are the hardened cells and lacks nutrients and they are in quiescent state, and how these fungus were able to survive, sir, in
I
nails.
didn't get the question properly. Sorry.
The nails are hardened cells, right, sir?
Hmm.
So if and their lack of nutrients also, and how these fungus were able to survive on it without any
Okay,
proper
that's a
nutrients.
nice question. But I told you the dermatophytes are keratinophilic. Keratinophilic meaning like like keratin. They are fond of keratin. So your hair, skin, and nails are rich in keratin. Okay. So because these fungi can colonize on keratin, they have the exoenzymes. Okay, they can secrete keratinases and then they can cause infection. But as you say, like the nails are much harder when compared to the skin. So not all dermatophytes can cause nail infections. There are certain species which are equipped, which can penetrate through the nail and they can cause infections. Yes, that was a
Thank you,
good
sir.
question.
And does steroid therapy causes immunosuppression?
Yes, yes. Yes, but the doctor usually will give a therapy which is the steroid therapy only when it is necessary. Because if you have other conditions like where it requires a steroid therapy, okay, they usually start with a mild steroid therapy. But depending on the if you have inflammatory conditions like rheumatic condition and all, sometimes the steroids are required because the pain will be more. So doctors will advise, but steroid medication has to be taken in consultation with the physician. Okay, and it is under the supervision of your physician. You are not supposed to take steroid therapy. And whenever you buy any cream, antifungal cream, or any cream from the market or in the medical shop, make sure that you go through the thing and you check whether they are free from steroids. Don't look in for creams which have steroids.
Yes. And I have one question, sir. One more question, sir. So does the fungi has the ability to cross the blood-brain barrier, sir, and it causes neurodegeneration?
But some of the fungi, like they can enter into the. I told you, like it's not the fungi, because we talk about these infections is like different ways of spread. Like, but fungi can spread to the meninges. Okay, I told you, like mucor can cause infection, rhinocerebral mucor mycosis, which is the infection of the central nervous system. Like Cryptococcus neoformans is another fungi, opportunity fungi, which can cause uh, cryptococcal meningitis. It affects the meninges. Okay, so it can cause infection. Yes.
And what is the best criteria required for a bathing soap against fungal infection?
Bathing soap actually, actually that doesn't. It's just to remove your. I mean, to remove your external dirt and all that. Fungal infections, like that. What I'm saying is, like, you need to be keeping yourself clean. Taking a shower every day is important. I don't know what sort of soap you use is not that important. But what you have to find is, like, you should find a soap which is not allergic to you. Okay, that's something which you need to check. And also it depends on your skin nature, because the people do not have the same sort of skin all over the body. Sometimes you have a dry skin all over the body, but your face will be oily. So what you require is soap which will clear your oil in the space. That should be something different. And what you use for your body will be different, because if you use the same thing which is going to remove the oil, okay, so you are already a person who has got a dry skin, then it's going to remove the remaining oil also, which is not good. So it's difficult, like to choosing the soap. And then if you are using some soap for a long time and you find it fine, then no need to change the soap. Just because something has come new in the market with a new perfume and all, no need to try that. If you are comfortable with the soap which is there in the market and you have used it for a long time, you are comfortable with it, then please continue with that. Okay, that's what they would say, like, because less perfumed and the less number of synthetic ingredients is more helpful. If at all you want to change the perfume, not necessary to you change your soap. You can change your the perfume, what do you use, okay, or your body spray.
And does this Candidiasis infection is associated with the COVID-19, sir?
That's what I told you. It's not just Candida. A lot of other fungi can also cause infection. Candida can cause severe blood infections called Candidemia, okay, which is the presence of Candida in the blood. Okay, normally Candida causes cutaneous or subcutaneous infection. Okay, so usually it infects the subcutaneous membranes. It don't go deep unless there is a compromisation of the immune system. But in COVID-19 patients, you also see that the immune system is lowered. Okay, so because of the lowered immune system, they have reported, like there are a lot of reports, not from India, from other countries, that there are fungal infections which are on the increase. But India also, like one report which I told you, is on the rise in the mucormycosis, rise of mucor infections. So yes, Candida can also cause infections in COVID people.
Yes, sir. Thank you very much, sir, for your wonderful explanations for all the queries that we have posted. And I hope the younger generations might be fascinated to work more on the fungal infections that are not solved at the mysteries behind the fungal infections through your wonderful lecture.
Thank
So I
you.
once again thank you, sir, for an informative lecture. And dear participants, kindly fill the feedback form which has been posted in the chat box. And the feedback form for the tomorrow session will be posted separately at the end of the technical session 4 in the evening. And we have come to the end of the 2-day international webinar on current scenario in therapeutics, prospects and challenges. Now I invite Dr. P. Tirumalivasan, Head, Department of Biotechnology, to deliver the vote of thanks.
A very good evening to all. It's my immense pleasure to propose vote of thanks in this 2-day international webinar on current scenario in therapeutics, prospects and challenges. First and foremost, I would like to express my sincere thanks to our college management for providing the guidance and support to organize this program. Next, I would like to express my gratitude to the research person, Dr. B.A. Vinmadi, Associate Professor, University of Malaysia, and Dr. S. Sagar, Senior Associate Professor, AIMS University, Nice University. And our special thanks to Mr. Isaac Parmat, Government of Visual Communication, Srimad Andavan. I also express my gratitude to all the staff members of Life Science for their excellent support. I thank non-teaching staff members of our college for the timely help. And last but not least, I thank the Almighty for making this one the first day program of a grand success. I thank you all for the opportunity. Thank you.
Thank you, sir. Thank you, sir. I once again thank the resource person for spending his valuable time and to share his wonderful knowledge with us. And I would also like to thank all the participants for your patience and active participation. Kindly join us the tomorrow session at 2:30 p.m. sharply. And kindly fill the feedback form which has been posted now in the chat box in the YouTube live stream. And then tomorrow's feedback form will be posted at the end of the 4th technical session by tomorrow evening. Thanks for your cooperation. Thank you all once again.